So with that, I'm going to introduce our first speaker. His talk is titled Genetics of CTNNB1 in the Context of Other Neurodevelopmental Disorders. Stephan Sanders, Professor of Pediatric Neurogenetics at University of Oxford. Welcome.
Thank you so much. Great pleasure to be here. All right, so fortunately, genetics has a reputation for being easy. Genetics is a complicated subject. I'm going to try and go through aspects here. I've done my best to a group where I'm meeting you for the first time to make some of it a little bit more accessible. There are a few bits which are very much orientated towards scientists. I don't even know which scientist I'm directing this towards. It's just there'll be someone in this room where it's important to. If there's a couple of slides here where they feel completely incomprehensible, that's fine. Okay, so with that slight caveat going, I'm going to have one more caveat. And this is just a list of the groups that I receive funding from. I don't think there's anything here which is directly relevant today, but I always show this. So my group works in Oxford. I've got Alicia here who's with us. We work on understanding why children get neurodevelopmental disorders. For many, many years, we've worked to try and identify the genes and the variants underlying it. We're now moving more towards the far end of understanding the biology and thinking how you use this information to make therapies. And in the middle, we are interested in how genes function, how they get regulated, and also how that differs by males versus females. And with all of this, we do it in a computational way. So we partner with other labs doing work to generate this data, and we try to make sense of it. When I started my career, this is the experiment which got me into it. We started off on the left with a family, in fact, many, many families, where we had both parents, a child with a disorder, in this case, autism, and a sibling who was a control who didn't have autism. We took their DNA and we did the test, which many of the families in this room will have had, of exome sequencing, back when it was brand new. And what we found was when we looked for germline de novo mutations, so these are variants which are not present in either parent, but are present in the child, so they're completely new, we found more of these in children with autism than children without. And that told us that some of these mutations were causing autism. And this is one of the important things about genetics. When you can show that a genetic factor is correlated with a symptom, you can make a comment about causality. You can say that that variant causes the syndrome. And this is one of the reasons why genetics is so powerful, is because we can talk about cause. But the problem was we didn't know which of these mutations. We knew as a group they were important. So the next insight came here. Here we're looking at some DNA. In blue, each one of those is one of the 20,000 genes in your genome. And at the top are the mutations we saw in the control patients. Each dot is a different individual, and there's no pattern here, they're just scattered. But here's the dots which we saw in the children with autism. And you can see that you've got some which are scattered, but some of them are in the same place. This is the signal by which we make statements that a gene causes developmental delay or autism or heart disease or epilepsy. And so the game from a statistical point of view becomes how many dots? Now, I could give you a few hours of lectures on that question, but we're going to skip to the answer, and that we found a way of doing this statistically. And so what we can do now is we can do this in large cohorts and find many, many genes which are causing symptoms. So these are the genes causing autism. And there you can see CTNNB1. Now, you could say, well, look, it's number 30 or something. Or you could say, there's 20,000 genes, this is right at the top. We can ask now a basic question, what do these genes do? And we find that broadly, there are two main things. The first one is that there are genes which regulate other genes, and CTNNB1 is one of those. The other thing we find is that there are genes which are important in how nerve cells work together. What is interesting is those two groups seem to cause very, very similar symptoms. And that might be a clue to how they're working. And that's something we're working on. But here's the first message. CTNNB1 is one of the most common group of genes causing these disorders. And so for all of these, for all of the ones in pink, we started with that first question of what are they regulating? And all of that information builds together. We've been able to go further than this and start to ask not just in autism, but can we identify genes which cause neurodevelopmental delay? There's so far 373 of them. So the size of the circle is an indication of how common that disorder is in children. Just to save you hunting, here's CTNNB1. This gene is one of the top players in neurodevelopmental delay. That is sad because it means that many people are affected. It is good because it means that we have a room with 82 families coming and attending. We have a family group which is emerging, scientists coming along. The higher on this list, the more activity there'll be, and the quicker and the more dramatic the race for a cure. So we're going to now move on to something a bit more specific about CTNNB1. We'll start off with some biology. I hope this is familiar from school. But on the left, DNA, which is found in every single one of our cells, makes RNA, which is single-stranded and doesn't last very long. And then that makes a protein. And we are basically built of proteins. This is called the central dogma and is mostly true. And for our purposes today, we can say it is true. So let's now try and apply that to CTNNB1. At the top, I am showing DNA. So we can take every single base pair inside you. So each of us has 3.3 billion base pairs of DNA. And each one of those has a number. We give them a chromosome, and then we give them a number, often in the millions. And that lets you find exactly one place out of those 3.3 billion. And just like you have Google Maps to find Ljubljana, we have the genome browser to go and take that and find it in the genome. At the bottom here, I'm showing the gene CTNNB1 in the RNA. We'll come to that in a second. Let's start with the DNA. So I talked about those coordinates. Here is a representation of a variant in the gene CTNNB1 in DNA space. So we have the chromosome, the position, and then the C changes to a T. And for many of you who've had a report, you will find a code like this on that report. This is probably the most accurate way of representing these variants. So let's move on to the RNA. So the DNA gets transcribed into RNA, and it's that RNA message which then goes to do something in the cell. There is a start, which we call the TSS. In genetics, we like to have special codes for everything, just to make it more accessible and easy. So the transcription start site, or TSS, will start. We have introns. Introns are bits which get made into RNA, but then get removed. You might wonder why. In fact, bacteria don't bother doing this at all. We'll come on to that later, but the simple answer is, if you want to build something complex like a human, you need more than 20,000 building blocks. And these introns give you much more diversity. We have the end. It has a special name as well, but we don't need to know it, so I'm going to skip over that one. And in the middle, we have the exons. So these little bits where there's rectangles, those are the bits which encode the protein. So the exons get spliced together. And so here we have our next representation of a variant. So this is called cDNA, or complementary DNA. There's the exons, and in red is the variant. It always starts with a C, a number, and then that same C to T change. And then lastly, those exons leave the nucleus of the cell to go and make a protein called beta-catenin. And here we have the third representation of a variant. And this starts with a P for protein. And here we change from one amino acid to another at position 90. And actually, as a rough estimate, if you take the number 268 and divide it by 3, you'll get about 90, because every 3 base pairs in RNA is one amino acid in a protein. All right. So let's start at the high level. What happens in CTNNB1 syndrome? So on the left, we have a typical individual who has 2 copies of the protein, or 2 copies of the gene making protein called beta-catenin, one from each parent. In CTNNB1 syndrome, we think for the majority of cases, the problem is that one of those bits of DNA is not working properly, making poor RNA, leading to no protein. We know from mouse experiments that if you're missing both copies, it's not compatible with life. And so this gives us our first answer of what needs to be achieved. We need to have more beta-catenin. I'm now going to move on to an experiment to dig into this gene a bit more. We're entering the realm of unpublished data, but that's fine for this group. Each one of these dots represents a tragedy. It represents a child or a fetus dying. But that family chose to give that tissue to medical research. And so we can look in those brains and ask how much RNA is made for every single gene. So this is some work done in my lab and at Yale, where we have a map of gene expression in the developing human brain. Alicia, who's sitting here, helped make that into an easier way to represent that data. And so here, these 4 tracks in black are showing how much RNA is made across the gene CTNNB1. And so the bottom 3 tracks represent pregnancy, so first, second, and third trimester. And the last track represents everything afterwards. Because from a developmental point of view, by about 1 year of age, things are fairly static. You will notice that these are higher where there are exons. And that's because RNA, we measure the exons. We measure it after the intron's been removed. The next thing to notice is that all 4 tracks look the same. This is not a gene which changes dramatically over time. And that's probably good. It makes it easier to study. We're going to add some more tracks at the bottom. So at the bottom here, we're going to talk about conservation. So this is conservation across species. So if you look at the very, very bottom there, we have zebrafish. So if I go and look inside the DNA of a zebrafish, I will find something which looks very, very much like CTNNB1. There's a track above, and so then we're going up between Xenopus tropicalis, frogs, chickens, elephants, dogs, mice, rhesus. We now have about 500 different species with this data, but these represent a big range across evolution. Above that is a track called Philo-P. This is a quick summary of how conserved each bit is. So why are we caring about this? Well, conservation is probably the best way we can tell which bit of DNA matters. You will see there is a dramatic peak in blue underneath each one of the exons. And that's because the exons are the bit that matter. If the exons are wrong, it's not CTNNB1. And so in genetics, we look at this conservation data a lot. And of course, this is driven by natural selection. Above that is a track which is talking about how the gene is expressed. And you'll see there's a lump there at the start, at the transcription start site. So this is meant to be a confusing slide. I don't understand this one yet, but this is work which needs to be done. I talked about introns and why they exist. Introns exist to give diversity so that one protein can do many different things. These are the known versions of that diversity. Now, at the top is the main one. The ones below are the ones which people suspect exists. This is an emerging area of science, but the main point here is it's not just one gene, it's one gene that makes many different forms of RNA. This is meant to be a bit more difficult, but someone in the room probably will need to hear this. This is looking at the expression of every single exon in CTNNB1 over time. The main thing to note from this is it doesn't change very much. And I want to give a contrast here. This is the gene SCN2A, another cause of neurodevelopmental delay, where it changes dramatically. And that's why we have the very yellow splodges on the right. So this is a negative finding. It doesn't change much. And the reason I put that in there is in the science world, we're going to have a debate about should we use an animal model like a mouse or a zebrafish or a rat, or should we be using a cellular model like human stem cells? And so here, I would say this is one piece of evidence that that cellular model is probably more useful than in other genes. Of course, there's no guarantee that downstream, the effects are not changing over development. On the downside, it means there's no obvious areas for therapy from a developmental standpoint. All right, so back to our gene here, CTNNB1. So I'm going to add in a second isoform. So this is something which is represented in the genome, which is a second start site or a second TSS. You can see that I've ringed it on the right here. Two things to notice. Firstly, in the top tracks showing where it's expressed, there's nothing there. And I've gone and checked, it's not our data. Other people have found the same thing. As best we can tell, this start site doesn't exist in RNA in the human brain. But here's what puzzles me. If you look at the conservation tract there, it is an incredibly conserved region. I have no idea what's going on with this. People thinking about therapy, is this an antisense? Is this an enhancer? Could this be a target for something about eRNAs? Don't know, but that needs more understanding. The next thing to notice, if you look carefully, you can see an extra little black bit where I've ringed it here between the two peaks. I'm going to zoom in on it. Okay, so here we've got the exon which we expect on the left, the exon we expect in the right. In the middle is a variable exon. This might be a target for an ASO. I have no extra data to say that it is, but if I was trying to think where would I start and try and make this gene go up, I would have a look at this because an ASO might be able to skip it or enhance it. And it looks like it removes 5 amino acids from the start of the protein. I don't know what that does, but it might be useful. And then at the end, here's another mystery. At the end of a gene, you're meant to see what's called a UTR. Again, we love our 3-letter acronyms in genetics, untranslated region. It's meant to be a single exon. And if it's not a single exon, that's really weird because it should make the RNA unstable. But when we zoom in, here's another mystery in this gene. There is a funny intron in this 3' UTR. That is really quite weird. It's so weird that I thought this is probably a mistake. But when we look at the tracks at the top, there's a fairly clear dip there, which makes me think there is something going on. And then I went to go and look at some long read data, courtesy of the lovely Jonathan Mills in the UK. And this intron in the 3' UTR is supported by long read RNA. This might be another therapeutic target. It is a very unusual thing to see in a gene. All right. So why did I highlight these 3 things? Really, that's for scientists thinking about therapies. We have a funny transcription start site, might be useful for CRISPR activation. We have a funny exon between coding exons 1 and 2 or normal exons 2 and 3, which might be useful for ASOs. We have a 3' UTR with a hole in it, which might also be useful for UTRs, for ASOs. Okay, so we've now looked at DNA, and we've looked at RNA. We're going to move on to protein. And this is work where Alicia, who's sitting there, has been incredibly helpful in putting this together. Alicia is about to start medical school. If you go and convince her that this is the gene to work on, you may have a doctor for the rest of your careers. All right, so we're now looking in protein space rather than RNA space. So there are 781 amino acids which make up the beta-catenin protein. We can number the exons for those, and that just helps us to transfer between RNA and protein. The first thing to notice is we have this black line called NMD escape. If you have so at the end of your RNA, there is a signal which says stop. And that's how the RNA knows when to stop making a protein. In many of the children with CTNNB1, there is a stop in the wrong place. It's in the middle. Now that stop leads you to having no protein, or it's meant to. There is an exception to that, and that the protein machinery can tolerate an extra stop signal in these bars on the far sides. And we'll see that that's borne out in the data. So earlier I showed the RNA exons, and this is just to show that those RNA exons map to this protein space. So in protein space, we can talk about protein structure. So here's an image of the beta-catenin protein. And you can see that there's these like spirals. Those are called alpha helices. And this is a protein which has, I think, 12 of those in a row. Yeah, 12. Those are represented here by the red bars underneath. And that's a prediction from an artificial intelligence model which uses machine learning methods to predict protein structure. One other thing to note is that these other brown areas here are where this protein is predicted to interact with other proteins. At the top, so we got very kindly sent the genetic data, and only the genetic data, for 125 individuals with a mutation in CTNNB1. And so this, at the time, I think represented every single individual we knew about. And so if you're a family here, there's a decent chance that one of these dots represents your child. We went through and firstly systematically made each of those be represented in the same code, so we could start talking about them as a group. And here we've plotted them across the protein. Now, let's go through what these mean. So we talked about, this is the P notation, for example, PQ193X. If it is a square, then that is an insertion deletion, which causes a frame shift event, and that's represented in red. If it is a triangle, that is a single nucleotide variant, which causes a premature stop codon, and that's represented as a red triangle. And if it is in yellow, that is a variant in a splice site, which leads to exon being skipped, which usually also leads to a premature stop codon. Or a simpler version of this, all of the variants in red and yellow are predicted to act in the same way, adding that extra stop signal in the middle of this protein. And we would predict that that not only leads to a small protein, but leads to no protein, because the cell tries to prevent that from happening. There are 4 individuals with missense variants. So a missense variant, instead of having an extra stop, one of those 781 amino acids has been changed to a different one. Sometimes that does nothing, sometimes it does everything. And the question here is, well, where are the rest? It is much, much more common to see a missense variant than a stop codon. And so my first reaction on seeing this data was there's very, very few missense variants. We've been doing some work in the lab. This is unpublished. Please feel free to take photos, but please don't share them in a public space, because there's a student who needs to publish this, and that would be a problem. We've been asking the question about this pattern of stop versus missense at scale. And this is a new approach which we've come up with. And so what we find is if you take 130 genes which cause developmental delay, some of them have more stop codons, like CTNNB1, some of them have more missense variants. We've created a new metric which talks about whether the protein is sensitive to these missense variants. The ones on the left are resistant. The ones on the right are sensitive. And we found that in the middle here, there's a group which acts mostly by missing one copy of the protein. And we found a group here which acts sometimes by missing one copy, but sometimes by having one amino acid change, which has a dramatic effect. And here's the weird one. Here's CTNNB1. The data from parents and children tells us that it is almost all stop codons. The data from how sensitive is the gene to missense variants tells us it should be incredibly sensitive. And so the question I have again is, where are all the missense variants? So I don't have a good answer to that, but I have some ideas. Actually, I've missed one important thing. See the blue bar on the far left? So CTNNB1 has also been associated with cancer. And the mechanism in cancer is a missense variant, particularly in this blue bar here. And what it does is it basically turns beta-catenin on and keeps it turned on. And that we call a gain of function. So that seems to be completely different from the patients which we're seeing a neurodevelopmental delay. And that gain of function, I think, is another important mechanism in this gene. So where are the missense variants? So one possibility is that they lead to a gain of function, and that therefore it's not survivable as a human. It's survivable as a cancer, but not as a human. That's one possibility. It could be that they lead to more severe symptoms and that they maybe leads to children who just don't survive to being born. It could be that it leads to milder symptoms and that we've just not yet recognized that there's a group of patients out there with the missense variants as well. And then a big question for us is having too much CTNNB1 the same as having a gain of function? Or, let me put that in slightly blunter terms, if I was going to do a gene replacement therapy, is there a risk of causing cancer? Which I think is an important question. This is a piece of evidence suggesting the answer is no. So I would start off with the statement that a gain of function, let me just go back here, this blue bar here, a gain of function is not the same as having too much. This is a hard concept and slide to interpret. And so this is one maybe orientated a bit more towards the scientist in the room. What we are looking at here is common variants in human DNA, which lead to changes in the expression of the gene CTNNB1 in different tissues, or for those who like the lingo, an EQTL from GTEx. What I notice here is that there's a dramatic range of expression of CTNNB1 in the brain. So what this tells me is that people tolerate quite a wide range of overexpression of this gene, making me think that it is more resistant to having too much, which is good news from a therapy point of view. The other thing which goes through my head looking at this, I've just said that almost every child has a mutation which causes a similar effect. And yet what I'm hearing from Laurent and Damjan is there's a lot of variability in the symptoms of the children affected, which is surprising given how similar the variants are. I wonder if this is why. Is it that there's varying levels of expression of this in the general population? And is that something we're going to see as we start to accumulate more data? Okay. Back to my stop codons. So the next thing I noticed in looking at this is that sometimes you see multiple of the same variant in the same place. Now, when I got into genetics, I was taught lightning never strikes twice. These are incredibly rare de novo mutations. They can happen anywhere in the genome. Why would you see the same one appearing twice? Well, since then, we've learned that there's actually a bit of code to this. So here is a representation of some DNA, and here are different types of mutations. Now, most mutations occur here and are incredibly rare. But there's a few mutations over here which are 10 times more common than the other mutations. And the time it happens is when you have a C next to a G. And the reason for that being that when this happens, you don't need to know the reason for that. It's just more common. And we call this CPG. And there's not that many CPGs in the genome because they are prone to mutation. But if I go back here, I can predict from the genetic sequence which variants should occur more often than chance. And I find these peaks here representing those CPGs. And if you look above each one of those, we see this tower of people with the same mutation. Okay? And so lightning does strike twice when you're a CPG. I should have that as a t-shirt. This makes sense in the data, but I'm not sure if anyone's got sharp eyes. This one doesn't. And this is the last mystery which I had looking at this data. I don't understand why that peak of individuals exists. So this is a mutation which is called PY333X. It should lead to the same symptoms as all the others, but the fact that there's a tower there, I can't explain it from the genetics, and I've really dug into this, which makes me think that it's more likely to be explained by the symptoms. And so if you are someone who has this, your child has this mutation, please let Laurent or one of the other clinicians know. We'll be interested in knowing if these children are in some way more severe, similar, different to the other children. And maybe there's a bit of a cluster going on here. Is it just 333X, or is it also a little bit more frequent here as well? I just don't know, but that would be between the numbers 310 and 340. Again, it'd be useful for us as clinicians and scientists to make sense of that. So why am I bringing this up? So I can't explain it, and I don't like things I can't explain. The other variants seem to be following the rules, that the stop codon is leading to less RNA and less protein. But for this one, it maybe isn't. Maybe it's doing something different. And we think about something called a dominant negative effect. So let me go back to this picture here. We expect to have 2 copies. In the kiddos who are affected, there's 1 copy missing. But there might be another state here, which we've not quite realized. And that's one where instead of leading to no protein, it leads to a smaller protein. Now, you might say, well, that's not a bad thing. And in some genes, it might not be. But the thing is that CTNNB1 interacts with lots of other proteins. And so this is a map of the proteins that CTNN binds with. And it does lots of different things. So we've got its role as a cadherin, its role as in sort of destruction over here. But up here, we've got its role as a gene regulator. And four of its friends, 4 of the things it binds to, are also genes which cause developmental delay. And 2 of them are really common causes of developmental delay. And so I wonder if this is what's happening, that this gene is binding to those. Let me just add a caveat here. This is speculation. This is absolute speculation. I don't have evidence to support this statement. But I wonder if what's going on is that it is binding to these proteins here, and that's how it normally functions. If you're missing 1 copy, well, that's a problem because not enough binding of these is going on. But if you have a truncated variant, it binds to them but doesn't use them properly. And so now you have less of these proteins, which are also sensitive to dosage, and therefore this dominant negative is sort of worse than having no protein at all. A possibility, and this is why we need to know more about the clinical symptoms. And I'm going to end with this and just come back to that first comment. This gene is not alone. This is one of many, many, many causes of developmental delay. And the types of therapies which we're talking about in this gene are the same as we're talking about in all the other ones. Last week, I was in a room with this gene here talking about gene replacement. We're at a very exciting time in medicine where these therapies like ASOs, gene replacement, genome editing are reprogrammable, which means that success in any one of these 373 disorders is directly relevant to success in CTNNB1. And similarly, your success is directly relevant to all the others. You're not alone forming links with these groups and working together. And as a computer scientist, we're trying to do this in the background to try and share that knowledge as well. I'm going to end by just thanking this very large group of people who I work with, but particularly highlighting Alicia, who's here, and of course, Laurent Servet, who's busy doing clinical assessments. Thank you very much.